Thanks. It's very nice to be here. You can see where I'm from. I'm going to talk a bit about life sciences stuff. I'm not going to go too deep into the research aspects, but also I want to touch on some of how our support for research is evolving in terms of cyber infrastructure or research computing and data. It's beyond just HPC, and you kind of need to be aware of these stuff. And I thought it'd be useful not to brag about what I've done, but kind of show you where I came from and what my background was. And kind of important in that is that I went to a bunch of these courses when I was a young graduate student on vectorization, on parallelization, that really kind of changed my career trajectory as a grad student, got me deep into computing. I had early access to computing in my bachelor's degree, which I did a double major in chemistry, math, computer science. We had a VAX at my college. And then I worked at Harvard doing programming early parallel machines. Was still interested in chemistry, went to grad school to combine chemistry and computers. There we were using the National Science Foundation centers in the US, computers like the Cray T3D, T3E, and so forth. And I started to go to these institutes. Eventually I became a teacher at some of these workshops, and they were really instrumental in getting me to where I was. At a postdoc, I moved to the NIH, and I was only able to use National Institute of Health resources there. We were using clusters of PCs, and IBM SP2 was available there. And then in 2000, I started my career as a research professor, not as a normal professor because I had a two-body problem. My wife got a job as a tenure track. I came in as a research track. Started to use local resources at our High Performance Computing Center. The Alliance Allocation Board became the TeraGrid, became Exceed, and eventually we had Blue Water as a big parallel resource. So I've used all of those, so many different parallel machines and GPUs. I was able to convert to the tenure track. At our first workshop for the International HPC Summer School at Sicily, I had just gotten tenure as an associate professor. In 2014, I became director of our Center for High Performance Computing. And the question is why a die-hard chemistry computational researcher suddenly got into supporting research computing. Happened because of my involvement in lots of different groups and advisory committees, from allocations boards, eventually advisory positions for Exceed, for other things, a whole list of acronyms you can see, and that kind of helped me be able to do both the research side and the computing side to some degree. Now, what do we do? Both Eric, who you'll hear later in a great software engineering talk, and my group are involved with developing some of the more widely used programs for simulating biomolecules with molecular dynamics. This is a time-averaged, smooth trajectory of a DNA simulation. You don't see the water and the ions that are there. If you look on the ends, occasionally you can see base pair opening, which you expect on a microsecond time scale. These were simulations run on the Anton machine, which is a special purpose machine for our field, by David Shaw. There's one at Pittsburgh that the community can use. And probably about 10 years ago, to run these 10 microsecond simulations on Anton took a few days, on normal machines took months, and now we're getting a microsecond a day on these kind of simulations on a V100 GPU, which is just awesome. And one of the things that we really focus on in my group is reproducibility. So if we do simulations with different initial conditions, we get the same results at the end. We want to see that we can converge the results. So in terms of like DNA, that's getting the same conformational distributions from independent simulations. We ultimately want to see agreement with the experiment and hopefully provide new insight. Takes a lot of computer time. I'm using about 1 million to 2 million core hours locally. On Exceed, until about a couple of years ago, I had about 10 million core hours a year in terms of an allocation. When I moved to Blue Waters, we were up to 12 million node hours of computer time a year. That's node, not core. That's like 2,000 GPUs a day for a year. It's a lot of computing power. It's very difficult to manage the data that results from that and the results. And running the simulations is easy. It's setting them up, figuring out what they mean, that's really, really hard. Kind of products that have evolved just over the past 5 years. We've released different versions of the Amber software from Amber 14 up to now we're at Amber 19, Amber Tools 19. One of the features of Amber that's very special is its GPU acceleration. We live all on the GPU, and we get performance for an equivalent generation of CPU and GPU about 10 times faster in the GPU than we do in the CPU, and that's much more acceleration than most codes, which give you on the order of 2 to 3 times speed up. We had to parallelize our analysis codes because we started doing ensemble calculations, and it generated so many files that it would tax the parallel file system if we didn't access the data in parallel. We developed multi-dimensional record exchange methods. I'll explain that in more detail. We have started the process of comparing results from different independent codes to show that they give the same thing, and that's kind of great for the community. We've shown that we can. If we can run on Anton or Amber or GROMACS or Charm and get equivalent simulation results, which is nice. We were able to re-refine structures. We figured out that we could go twice as fast in the simulation by changing the integration time step by shifting hydrogen mass. We showed for the first time reproducibility and convergence, and spent a lot of time assessing all the force fields, validating them, optimizing them, and you'll see in some cases proving they work great, in other cases they don't work at all. So what does this research require? Computing support. This is kind of physical and people resources, both locally and nationally, to help. Just to use a machine like Blue Waters is not necessarily straightforward. There's problems that arise, so it's nice to have a local liaison at that center that you can interact with and can help you with the problems. And it's not just access to the compute, it's how do we use that compute most effectively. And I just put kind of what's in our center. We used to be a high-performance computing center. About a decade ago, we had a machine like an IBM SP2, and we help users run on that. Now we support research computing and data broadly defined, and that's things like virtual machines, cloud, security, data compliance, protected environments for mining restricted data, advanced networking and data movement. And you really need a group of people that can help you understand what the capabilities are. So you as a graduate student or postdoc or young faculty member can get advice from experts to help you how to effectively use these resources. And I kind of put up my definition of research computing and data from the Campus Research Computing Consortium, which I work with. Really, it's not just HPC, it's all sorts of this other stuff now that you kind of need to be aware of, and it's going to become more and more pervasive. And this is a challenge, you know, beyond learning about MPI and OpenMP, we're going to have to learn about how to deal with FPGAs and different memory hierarchies that are emerging and all sorts of new, you know, cloud stuff. It's very different on Amazon than it is on Google for doing your workflows. Kind of stuff beyond data or beyond compute that's important. The research data management, compliance, movement, and stuff is really hard. You've heard a lot about analytics and machine learning. You hear a lot more about that's becoming pervasive. When we first did the summer school, we didn't even mention the words analytics or machine learning, and they started coming in about 5 years ago, and now it's a much bigger part of our efforts. How you do the workflow, how you manage your stage simulations and do this, kind of like the packing we heard in the last talk of trying to optimize fitting the different sized and timed workloads into the same space. Provenance, how do you remember what you did in the simulations? High throughput computing, gateways, containers, first modules, cloud, Jupyter, all this stuff, software engineering practices we'll hear about from Eric coming up soon. Python is emerging as kind of the de facto code for rapidly prototyping your scientific application, and it's awesome. It has all these different libraries, ties to machine learning, ties to numerical libraries. It's not very performant. Our C++ code for analysis is between 2 and 10 times faster than any of the Python equivalents. And so if you do the Python right and optimize the portions that need to, it can be very, very effective. I'm still kind of old, so I'm more in the Perl era than the Python era, so I haven't switched over, but my people have. We have to worry more about accelerators, emerging memory hierarchies, and viz. Okay, so in general, for any kind of good computational simulation experiment, kind of what do we need to do? I'm going to talk about here in the context of converged simulations of nucleic acids. One is the initial conditions in all our cases are very, very important. How do you set up the initial models, the parameters? How do you kind of relax the system before you enter into what you call production simulation? And usually it's good to have multiple initial conditions so you can test they go to the same place. When I first did some simulations of RNA a few years back, about a decade ago, of this particular system, it's time average and smooth. It transitioned to an alternative structure, and that's really nice, except the structure wasn't real. It doesn't exist experimentally, so it was a complete artifact. And we had started with the two structures on the ends, these NMR structures that were in the protein data bank, multiple initial models. And it turns out these models weren't that stable. They were old, refined methods for generating them. When we re-refine these structures using the original experimental data, it turns out these two different structures are actually very similar. The only difference is a mismatch in the RNA in the center of the duplex here. And now, once we've done the simulation with the experimental restraints, if we turn off the experimental restraints, we can now get good fidelity modeling of those structures. Another example is here. You can see the original NMR structure kind of shows a spaghetti or a range of structures that fit the data. When we re-refined it, we got a much tighter structure, and we were able actually to induce magnesium-dependent conformational changes, which turns out when magnesium is not present, it's adopting multiple conformations, and the magnesium traps one, and we're able to reproduce that, which is really kind of cool in the simulation. Once we get to production MD, we've moved to a model of, as the plasma physics have, of kind of ensemble calculations or multiple independent runs. And in our case, application of methods to speed up the conformational sampling. So ensembles or replica exchange or multidimensional replica exchange, where we exchange information periodically between independent simulations to kind of accelerate how it's moving through space. So kind of shown here is my Amber engines. We're running a bunch of independent simulations, and they exchange information periodically. And we've proven this can generate the right ensembles and significantly speed up how we sample the conformational space. How do we do this? Well, you heard about yesterday, if you were in the MPI track, these communicators. MPI Com World is everyone can talk to everybody. So we can set up some different ones. So in the world, I can talk to any one of the different instances, but I also want to have communication within each independent simulation run. That's what I'm calling my Com Sander with the program Sander. And then occasionally when I'm going to exchange information between the independent simulations, I need the masters on each one to talk to each other. And so this was really easy because we have the existing code base for the molecular dynamics engine. All I have to do is put a little wrapper on top, change MPI Com World in the main engine to be now Com Sander. Outside, I'll have the world and the ability to talk between the masters, and I can exchange the information. And it's really kind of simplifies things. So I put kind of the code and how you do that for you for future reference. Will be on the web page or in the video. Essentially, we kind of set up these different communicators with this MPI Com split command, and allows us to kind of easily do this, which is kind of very nice. The drawback of this is, though, which is being more addressed with the plasma physics guys, is if one of my MPI processes dies, the whole thing goes. And I have to know exactly how many MPI processes I'm going to run because I've got to fit into the queue in that particular place. I can't just dynamically increase and decrease it, which is where we're trying to move to now, to have more asynchronous ensembles that can backfill in. So using long simulations or these methods, we can converge the conformational structures of a number of different systems, like duplexes. You know, back in 2007, the state of the art was a 1 microsecond simulation, and it looked stable, and you could see certain properties. And the way we think about structures is we would average over a time window of like 5 nanoseconds and compare the structures. This is between two different, Anton run and regular run. And that's the way we were used to looking at structures, and it turns out we weren't really running towards convergence at this particular time. Here's an example of looking at salt effects on DNA structure. And you might think there's differences between these structures at different salt concentrations. All the publications up till this year, the longest that have been done, were 1 microsecond simulations looking at salt dependence. And I could calculate RMSDs, and we could argue about the statistical significance of the difference of these things, but they're just an artifact of not being converged. When we run it out to the 10 microsecond time scale, now all the structures look virtually identical. The differences are on the end where there's differences in base pair fraying. And we show there's no influence of salt with the current force fields and some kind of convergence. There's a nice paper by Bernard Knapp that talks about this. If you run a single simulation and you try to interpret it, the results can be entirely misleading because you're going to over-interpret it. You may have missed things unless you can prove that you've converged the sampling. Much better is to run lots of independent simulations and aggregate the results and see what's up, and that's a better way to do things. Now, I showed those kind of spaghetti structures a few minutes ago where you kind of saw the blurring in them. Once we got to Anton machines and GPUs, we could now run much longer simulations, and it turns out for DNA in the Watson-Crick form, if you average over longer time windows, like 3 microseconds, the structures are identical over different windows from the simulation. And that picture is of 5 overlapping structures. You wouldn't think there's 5 overlapping structures. You can see a little bit of blurring in the end where there's a little differences to the base pair frame, but internally, the structures are identical. First, we thought that was artifact, but it turns out to be real. And it's amazing that we can get agreement with experiment for a DNA duplex with the current force fields like the Barcelona one or the OL15 and particular water models that are less than half an angstrom away from the average of the NMR structures. That's just truly phenomenal in terms of the reliability, particularly for a force field that wasn't fit to a helix or a structure was fit to a residue and just generalized. Okay. How do you know you're done in the simulations, and this is going to be true for all simulations one runs, is you want to assess this convergence. And in our case, because we're looking at time average or structure as a function of time, we want to look at properties, both the structure and the dynamics, and kind of measure what's happening. And so the way we've done this, starting about 2013, was we would take independent runs, we aggregate them, we put them together, and then we calculate properties. And then we separate the data again and we project those properties. So like principal modes of motion or cluster populations. The reason why you want to put the data together first when you're calculating the clusters is because if you do it independently, how do you know cluster 0 is the same as cluster 0 in the separate run? You don't. So if you do them together, cluster 0 is a separate, same definition. And when you separate it, you can see if the populations are the same in both. And this can be done with the RCPP Traj code that's freely available and works with any of the simulation packages, GROMACS and others. Here's an example of a projection of the first 5 modes of motion of this DNA duplex. There's a dotted line and a solid line, and blown up here, and they're essentially identical. So the motions that we're seeing in these 2 independent trajectories were exactly the same, suggesting we've converged for those properties in that particular time scale. Here's showing the Kullback-Leibler divergence in the modes of motion, either when we omit the termini, here, or if we include the termini. If we include the termini, we see there's good convergence, but then one diverges away. That's because a base pair opening event occurred in one trajectory, but not the others. And then later here was a 2 base pair opening event that occurred in one trajectory, didn't occur in the other, so we had a larger divergence. And so these are really nice ways to kind of assess if you're converged or not. We can also get cluster populations, same thing. Shorter simulations on top, there's overlapping lines that are crossing, so the population is changing. In the longer simulations, there's no crossing anymore. We've got a converged set of populations. Okay, so I want to kind of, one of the issues we have our ensemble calculations. We're exchanging information between them. So I'd still have to write out my data that kind of gives my current state or my structure as a function of time. Do I write it out in temperature space? Do I write it out in replica space, which is just the ensemble instance, like ensemble 0 instance, trajectory versus someone? Or do I write it out in Hamiltonian difference? Well, effectively, I have to sort it in all possible ways, and this is why we had to parallelize the analysis code, because we wrote it out in one way, but we had to see it in all other possible ways. This figure is showing a population for each independent ensemble instance in 3 separate simulations, kind of how far the population of structures away from A-form reference. So what you should see is that every ensemble instance should have the same profile. They don't. You can see like 3 of these simulations have conformations that are highly populated here that the other ones don't have. And if I had just looked in temperature space or looked in another space, I wouldn't have known there was these differences. So we need kind of multiple ways to look at this data. So what we have in the analysis codes now are, We can do MPI across files. We can do MPI across ensemble instances. We do OpenMP for time-consuming tasks, like pairwise distance calculations. We do CUDA. And so what does this mean you should learn? Should you learn MPI? Should you learn accelerators? Should you learn OpenMP? Yes, all of these things. They all may come into play as you evolve your code, and it has different use cases and usage needs. We now have Python interface to our optimized C++ code because a lot of people will still want to work in that. And we're constantly adding new features. For example, longer and longer trajectories, you can't load the whole thing into memory, so we've got to do data caching to disk, so we had to kind of implement that type of stuff. Other properties that are shown there. Paper on the parallelization of CB-Traj came out last year. A quick word on why I haven't talked about RNA yet, but it turns out RNA spectacularly blows up in almost every simulation if you run it long enough. If you run it short enough, you'll well reproduce experimental properties if you start with the experimental structure. But invariably, if you go longer and longer, it will blow up and go to a new state. Why? The DNA helix is beautiful. It reproduces the structure. The RNA blows up. It has to do with this issue of balance. And it turns out, if you think about DNA, if I'm hydrogen bonding a little bit too strong or too weak, or if I'm stacking a little bit too strong or too weak, it doesn't change the helical structure at all. It just changes the stability, the melting temperature, and these kind of things. But in the case of this RNA, if I'm overstabilizing a particular set of interactions, if I don't have the balance just right, I may overpopulate this conformation compared to what I would expect to experiment. And that's what is happening. It turns out for systems like proteins, where an alpha helix buries all those hydrogen bonds and beta sheet buries all those hydrogen bonds, the balance isn't as important. Whereas important is for these subtle loop-loop interactions at drug binding sites. There it's critical, and the same thing with RNA and DNA. Duplexes are fine, but if you go to anything more rich with these weird alternative structures, we tend to get the populations wrong with every available force field. Here's another example. Okay, how to validate the simulation results. This is really tricky. So say I'm simulating an RNA tetraloop and it deviates from the experimental structure. What should the population of those anomalous structures be? Should it be 0? Should it be 5%? Should it be 10%? Well, if it's more than 10%, we'd see it in the experiment. We'd see it in NMR. But is it 7? Is it 5? And these are kind of the questions we're trying to get at now is what are these kind of populations for simpler RNA systems so we can further validate these force fields. Here's my workflow for my latest grant that's just going to get funded, which is really exciting. It's not just about running my high performance computing calculation. It's about comparing to experiment. It's about optimization to improve the force fields, probably machine learning in there. It's optimizing the analysis codes, so eventually I can steer the HPC codes on the fly to do more sampling. It's comparing to experiment, doing max end-to-end. So we're constantly adding to our repertoire and code base and requirements to kind of satisfy the problems we're trying to solve. And this is just one aspect of life sciences research. There's a whole bunch of others as well in terms of cryo-EM, genomics, and other areas. Okay, I went a little fast, but I didn't want to go over because I didn't want to have you miss your break. So my final thoughts, and some of these will be echoed by Eric when he gives his talk coming up soon. For me and my lab, the rate limiting step isn't running our simulations anymore. Setting them up is a pain, getting them right. Run them, we just let them go. We just let them run, and we run them for weeks to months. But figuring out what they mean, doing the analysis is really the rate limiting step, as is the data management, moving it from the supercomputer center, processing it in various different ways, transferring it home. We can't do all the analysis at the supercomputer center because we don't necessarily know what we want to look at yet, what properties. If we knew all the properties to analyze, it'd be easy, but we don't. If you make your data available, it may be reused. So we've done this with the simulation of the Anton results and a lot of our data now. All my papers and the supporting information have kind of the scripts on how to do the analysis, and we put the trajectories out there and don't even really advertise them. People are actually using them and publishing stuff based on the data that's available, which is kind of cool. But this is a huge challenge nationally, internationally, or at our local institutions. How can the local institutions or governments provide the resources to let us have our data available that people will use? Which data is the most important if you only have finite resources? Who's going to help you curate that data, manage that data? How long do you keep it for? These are big challenges that we're trying to figure out through a lot of different communities. Again, it's not only about the HPC, it's about the entire workflow and research process. I think Eric might talk about this. Code development optimization never stops. Amber was developed probably in the late 70s or early 80s. It's still Fortran, but it's evolved. It's highly optimized Fortran. It's got CUDA in there. It's got C. It's got C++. And it's continually changing, but not that fast. If code development and optimization stops, often the codes die or fail or go away. Practice good software engineering practices and good code development. Being able to code increases potential career opportunities. If you learn about the basics of cyber infrastructure, research, computing, and data in multiple of these areas, like cloud or containers and anything, you can get a job. I mean, if you can spell Kubernetes, you can get hired in a DevOps position out in industry. And these are great alternative careers from becoming a professor, because not all of us can become professors. Some are going to not. And learning this stuff's good. But you know, I did a 7-year PhD. I spent 2 years coding. I didn't get any papers for that, and I learned a lot. But it's a different path. GPUs, OpenACC versus CUDA versus multi-node, and now new emerging accelerators. We found that CUDA's been awesome for getting the performance of Amber to be 10 times the CPU. We never could do that with OpenACC, but CUDA is difficult. Every new card that comes out from NVIDIA, we have to re-optimize things because they change memory speeds, ratios of memory, they drop instructions, and it's a real difficulty. And there's only a few people in the world that can actually do it, touch the code. We only have, in both GROMACS and in Amber, there's 2 people, maybe, 2 to 3, that can really well touch the CUDA code. Convergence, reproducibility, reliability, validation are important for any simulation experiment in any field. Collaboration helps. It's good to be collaborative. Source code and parameters should be publicly available. Machines do not evolve to improve our performance. We need to evolve the algorithms to the machines that are given to us. And this can be very painful. GROMACS spent a lot of effort optimizing for Knight's Landing. Amber spent a lot of efforts optimizing for Knight's Landing, and then they killed it. It did make our regular parallel code faster on CPUs, but there was a lot of effort that went in. And I worry now about, you know, optimization for NVIDIA GPUs, because you look at the Department of Energy roadmap towards the exascale, none of the upcoming machines include NVIDIA GPUs. They're all AMD GPUs, which we don't have any code for. And it's really scary. So those big machines are all without the NVIDIA. What are we going to do with our NVIDIA code? We'll run them on commodity cards, so we'll still be okay. I will stop there and let you ask questions if there's any.
Yeah, thanks a lot, Tom, for your nice presentation. So.